Structure and function of protein. Sulfur containing amino acids are cysteine and methionine. Heme in hemoglobin is in the hydrophobic pocket. Blood group antigens belong to the class conjugated proteins. Fluorasamine is used to detect amino acid. Acidic amino acids are none of these. Basic amino acids are arginine and lysine. Acidic are asparagine and so basic is arginine and lysine. Aromatic amino acids are tryptophan, tyrosine and phenylalanine. Ninhydrin is a reagent used in detection of alpha amino acid. Non-essential amino acid is tyrosine. What is essential is phenylalanine, lysine and threonine. Alpha helix structure H bond within peptide. Same charge 2 protein separation is done by Cephadx. Iron in the hemoglobin is held by polar bonds. Vibrational property of protein is due to tertiary structure. Tertiary structure of protein is maintained by all except, so it is maintained by H2 bond, hydrophobic bond, ionic bond and disulfide bond. The color of cheloglobin is green. Sulfhemoglobin has a dirty green color. Siderophylline is beta globulin. Helical content of polypeptide can be estimated from optical rotatory. In determining protein structure, performic acid is used to oxidize disulfide bond. In electrophoresis, if the pH is above isoelectric point, a protein will migrate to anode. Which of the following is correct about breakdown of hemoglobin? Hb to heme, then to biliverdin, then bilirubin, then urobilinogen. Porphyrin is formed by joining together 4 pyrrole rings by means of bridges methinyl. Which of the following is a derived protein? Peptones. Monoclonal electrophoresis is useful in the diagnosis of multiple myeloma. Siderophylline is transferrin. Which of the following is not a part of hemoglobin molecule? Ferric ion is not a part of hemoglobin molecule. Which are the parts? Pyrrole rings, vinyl group and histidine. The Klett-Summerson's photoelectric calorimeter is used for hemoglobin estimation and the reagent used for this is Drabkin's reagent. Aromatic ring is present in phenylalanine. What is the average number of high energy bonds required for the synthesis of one peptide bond? 4. Bohr effect is effect of PCO2 on oxyhemoglobin dissociation. Heme in hemoglobin is surrounded by non-polar environment. Collagen is unique in that it contains much hydroxyproline and lysine. The most abundant protein in the human body is collagen. Ninhydrin reaction gives a purple color and evolves CO2 with asparagine. Collagen is a major component of bone. Flexibility of protein chain is determined by glycine. Myoglobin has an O2 dissociation curve that is unaffected by wide range of pH. Quaternary structure of protein is the interrelation and arrangement of polypeptide in a protein with more than 2 polypeptide chain. Structure of protein is best studied by X-ray crystallography. Iron is complexed in hemoglobin to histidine. Amino acid which doesn't participate in alpha helix formation is proline. The amino acid whose ionization is maximum affected by changes in local and microenvironment is histidine. The integral membrane glycoprotein of human RBC is glycophorin. In terms of chemical structure, which one of the following is a tetrameric glycoprotein? Insulin receptors. The coiling and folding of protein requires sulfide bonds. False statement about hemoglobin structure. Hb has 2 polypeptide and Hb is structurally similar to myoglobin. Both are false. So what is true is iron is present in ferrous state and ferrous ions are in porphyrin rings. Indole ring is present in tryptophan. Triple helix is found in collagen. Albumins and globulins are simple proteins. Regarding isoelectric pH, true is net positive and negative on molecule are equal. Which is a denaturating agent? Guanidine. Electrophoresis done under pH gradient is isoelectric. Most abundant collagen in body is type 1. At pH 7, the binding of 2.3 diphosphoglycerate to hemoglobin occurs at which site? Amino terminal. Keratin in the nail is exceptionally stronger than others because of disulfide bond. Phospholambin is a protein that leads to sequestration of calcium in endoplasmic reticulum. When the following amino acids are separated by running them on agarose gel at pH 7, which one of them will migrate slowest to anodic and lysine? Molecular separation of two proteins with same charge can be done by gel diffusion chromatography. Intact peptide bond is necessary for which test? Ninhydrin. The best method to differentiate protein is by gel chromatography. Defect in collagen formation is seen in scurvy and osteogenesis imperfecta. Optical transmission through a solution depends on concentration and path length. Which of the following statement characterized both hemoglobin and myoglobin? Hema at hydrophobic pockets. At isoelectric pH, protein have negative charge 0, don't migrate, and at isoelectric pH, protein don't migrate. Protein are separated on the basis of size, SDS page. Proteins can be separated by the following except none. So proteins can be separated by electrophoresis, ultracentrifugation, gas chromatography, and salt separation. The following separation technique depends on the molecular size of the protein: gel filtration chromatography. The conversion of optically pure isomer enantiomer into a mixture of equal amount of both dextro and levo form is called racemization. The collagen triple helix structure is not found in cytoplasm. Protein acts as buffer due to which property? Amphipathic, that is amphoteric in nature. Alpha helix and beta pleated sheet are examples of secondary structure. Cytochrome C of the bacteria has 50% identity of amino acid sequence with that of human. Which of the following is the most conserved parameter in these two proteins? Tertiary structure. Copper is required in collagen synthesis for lysyl oxidase. Which one of the following is not true for alpha helix? It has a net dipole moment. Which of the following amino acids have side chains that are negatively charged under physiological condition? Aspartic acid. One of the following is an example of phosphoprotein, casein. Which of the following can be a homologous substitution for valine in hemoglobin? Glutamic acid. Biological value of protein is related to protein quality. The molecular weight of a protein can be determined by sodium dodecyl sulfate H page. In chromatography, mass movements of the substances is due to diffusion. Zwitter ions have positive ions is equal to negative ion. At physiological pH, the carboxyl terminal of a peptide is negatively charged. Substitution of which one of the following amino acid in place of alanine would increase the absorbance of protein at 280 nanometer? Tryptophan. Secretory proteins are synthesized in endoplasmic reticulum. Which one of the following can be a homologous substitution for isoleucine in a protein sequence? Valine. Proteins are linear polymers of amino acid. They fold into compact structures. Sometimes these folded structures associate to form homo or heterodimers. Which one of the following refers to this associated form? Quaternary structure. The structural proteins are involved in maintaining the shape of a cell or in the formation of matrices in the body. The shape of these proteins is fibrous. Protein purification and separation can be done by all except densitometry. Protein that precipitates on heating to 45 degrees Celsius and redissolves on boiling is Bence-Jones protein. Hemoglobin electrophoresis is based on charge. In collagen synthesis, hydroxyproline is formed from proline. Lipids and proteins interact in membrane by hydrophobic interactions and H bonds. Which of the following is extracellular matrix protein? Collagen, laminin, and elastin. Basic amino acids are arginine, lysine, and histidine. Folding protein are calnexin and protein disulfide isomerase. Methods to determine protein structure are X-ray crystallography, NMR spectroscopy, and infrared spectroscopy. Which of the following has maximum buffering capacity? Histidine. Which component transfers 4 protons? NADH-Q oxidoreductase and cytochrome Q oxidoreductase. Hydrophobic amino acids are methionine, isoleucine and alanine. Trypsin cleaves arginine and lysine.